Okay, the overview of this course is that we have it divided in 3 phases. And that is because we look at it as a process or a progression of you learning and doing science. So the first phase, which is the longest, is the introductory phase, and that is you learning how to do science. And so we're going to be doing a lot of what we call guided inquiries. And what that means is I get to ask the question for the day. But then when we move into phase 2 and 3, we do open inquiries, and you get to ask the question for the day, you as a team. And each team will be doing a different thing and asking a different question. So it's really a lot of fun. And so what we're talking about are things like observing, which we will do today, designing an investigation, which you will do today, doing a prediction, which you will do today, measuring and communicating. That's important. We communicate almost every lab. I will have you communicate as a team and so forth. So, you know, collecting data, analyzing, that's the kind of process skills we'll be working on, as well as learning the tech components in the classroom, because as you can see, we have a lot of technology. And so in the room, I'm a facilitator really, not an instructor. So I'm just one of you, and I'm going to be helping you do some science. That's our kind of philosophy in the classroom. Alrighty? Okay, let's go back then to our course folder here. And this tells you then where you're going to be putting your work. Okay, so Team A, your work goes in Team A's folder and so forth. Now, to get the template for your Team Lab notebook, you have to go to the course folder and click on templates. Okay, everybody with me? And when you click on templates, you will see there are the first 5 labs listed. What we need to do is open up Team Lab Notebook 1. That is what we are going to complete today.
Therefore, concluding which one is ortholess water. Scrap it down. What? I'm looking at the next question.
Open up JUBI. JUBI stands for the Journal of Undergraduate Biological Inquiry. And part of your team grade is to make 3 entries into that journal. It's an electronic journal, and so you will be putting a little journal article in there on the investigation you have done. So yes, we are really doing science. We're writing little articles, we're presenting them, we're talking about them, we're sharing them with the class. And the last JUBI, JUBI 1 is a little mini, getting you to learn. JUBI 2 is a little more difficult. And JUBI 3 is, voila, the presentation one, where you get to do a PowerPoint or a poster. And your team can decide this as you get there. In this course, as you can already guess, by the way you're seated, we work in teams. It's a team approach the whole semester. And so the first thing I need to do is put you into teams. And to do that, we're going to play a game. Okay? And so what you need to do for the game is use any kind of communication you want except voice. Okay? And the game is such that I want you to line yourselves up as a class so that the January birthdays are over here and the December birthdays are way down here. Okay? And just leave your things where they are. Okay, so what I want you to do then is, we'll start with the January people here. I want you to introduce yourself, use the name you like to be used, if, Thomas or Tom, whatever it is, and tell us your birthday, and tell us if you're, freshman, sophomore, where you are in your educational career. So if you want to start.
My name is Mara Gibson. My birthday is January 13th, and I'm a junior.
I'm going to sort you in teams, simply by my birthday lineup in 4s. Okay, so you may choose anywhere in the room. There's 6 teams, and they're designated. If you take a look at the computer screen, there's A, B, C, D, E, and F. So it doesn't matter to me. Move your things and join your new group. So, okay, here we go. 1, 2, 3, 4, and there's my first team. Your team is very critical. And so in the room, I'm a facilitator really, not an instructor. So I'm just one of you, and I'm going to be helping you do some science. That's our kind of philosophy in the classroom, alrighty? Now, we work in cooperative groups, and I already have you in a team, and I hope your team becomes cooperative. And to begin that journey, we have a lunch bag that is by your place. And no, it doesn't have lunch in it, unfortunately, but in it there are 4, and you'll see them if you take them out. You're supposed to choose one of the 4 cards in there, okay? And the cards designate a role that you might have in your cooperative group. Okay, and in the lab manual as well, it explains the roles. Okay, so whichever you choose, you can see you have group leader, you have observer, you have the manager, a material manager, and then you have the encourager and also the recorder. And the recorder is usually known as the mouse hog. So we try to change roles per lab so that nobody becomes really the mouse hog.
Prokaryotic and eukaryotic have to do with the rest of it.
Prokaryotic are cells with no nucleus, and then the eukaryotes have a membrane-bound nucleus. So like the difference, it's just...
So is that another way to distinguish?
Yeah, that's how they split them up. They split them up for cell type, oxygen requirements.
Organism, reproduction.
Yeah, reproductive stuff, and then what they eat.
So you guys did, you know,
It's probably not 3 again.
the first activity is a brainstorming activity, kind of an engagement, getting your brain thinking. And what I want the teams to do is start on their team lab notebook, enter your names, enter the date, go to activity 1, and each member of the team is to contribute 3 thoughts on the role of biology, the role of water in a biological system.
It's responsible for different climate control and distributing water to plants.
Also, where it says human survival, it's also plant survival and like all life survival. So I don't think it's just humans.
We're moving right into our next activity, which is the inquiry activity. And we're going to use water today. That's why we had to make you think about water in our experiment. And our experiment is based on these 3 paper towels that are sitting in the middle of the room. And the question I get to ask, because it's a guided inquiry, and the question to the research teams, that's what you are now, is to find out which one of those 3 paper towels is the most absorbent.
Yeah, it's not holding anymore. And that's only 3 millimeters. It's gonna sit in this little thing like that. Cool.
That'll work.
Ready?
Ready. Yeah, you know what? Because if it is going to be a good absorber, it was not going to spread as much, right?
Yeah.
You'll have some data to tell us about.
Oh boy.
Wow, we have a clear winner.
The plane weight is about 24 milliliters, right? Oh, what a disappointment. That's because it was bigger.
So what we did was we took 25 milliliters of water and we just poured it on the table because we love those Bounty commercials where it just soaks it up. So we poured 25 milliliters on the table and we took just a square of each paper towel, set it on top for about a minute, and what we did at the end of the minute then was would we'd pick it up and see which had left the most water on the table. And so we kind of determined that that would be our most absorbent, whichever one left the least amount of water on the table after about a minute.
All right, the question, of course, was which paper towel would absorb the most water. And the method we employed was to take this pink cup full of water and to submerge each paper towel one at a time for 10 seconds. And then we kind of let it drip. We took it out and let it drip for 3 seconds just to let some of the excess run off. And then we squeezed the paper towel into the graduated the beaker and measured how much water came out of the paper towel. So we did that 3 times, and well,
We came up with the Health and Happiness held 25 milliliters, the plain white was 24, and the strawberry was 51. So obviously that held the most.
We predicted that the plain white paper towel with the roses would hold the most, but our prediction was wrong.
And then we realized at the end that the strawberry one had 2 layers. So then we realized that that was probably the reason why it was twice as much as the other ones. Plus it was bigger in size, so more to absorb.
And if we had to do something differently, we'd probably run the experiment more times to get it more accurate. And maybe run something different just because we drop a little bit of water.
Did somebody do a correct experiment and someone did a wrong experiment? What do you think? Team F, I'll ask you first. What do you think about that?
I don't think everyone did it wrong one or right one. Everyone had their own ideas of which way to get the best conclusion.
Okay. And so what did everyone base their conclusions on? Team B. What component part is that? What part did you have to show us? The data or the evidence that says this is what my method produced. Let me ask this one. Would everyone in the room do an experiment differently if I had you do it now again? So did you come up with a whole lot of different ideas of how one might do it? Okay. And so this is kind of how science moves, is it not? It's exactly how science moves. Somebody does this, another lab does that, somebody comes up with this data, somebody tries something else, and we either are going to dispute it and say no, or we're going to say, yeah, we're going to accept this and try a different method. No, questions that are going to be testable. Because you're going to be actually setting up an experiment. So you have to think, can I do this? Because some questions are not testable, at least in our setting, you know, and they're better done by answering by research. Looking it up, somebody's already done it, why do it? Okay? Yeah, right? That's true.
Chose a proper amount of water, and our team's question was talking about we were going to experiment with overwatering. Is that I mean, like, our team's question, our team's prediction, are those feasible things?
Well, what do you think? Do you have a reason you have selected that? Because
it's feasible to do in here. I don't really know if we had to do it.
Oh yes, we are actually going to do this experiment, yes.
What like the proper amount? Okay, let's say like, I can't do anything on the proper amount of CO2, right? Like we're gonna cut down the CO2 content in the room. I mean, so we just want to do something that was feasible and acceptable weather. I don't want to thermostat. Stuff like that.
Boy, and maybe you can't change the thermostat, but you can do other things in the room. We got lots of things in the room.
Okay, so...
Right, we got water baths, we got those growing chambers over there. We got lots of things in the room.
Okay, so, but what about the overwatering? Is that feasible, or is that just not, should we just go with temperature?
Well, what I'm saying to you is that similar to putting Lysol on your plants? That is, we know that this is going to kill them. What do you think about overwatering? What's your prediction? Because?
Here's the concept map for the introduction.
Hang on 1 second, we're cooking with gas. Okay, so if we overwater, the plant will die. So we just want to do an experiment where we're positive, we don't know the outcome.
You want to do something that is going to be something that you aren't sure what the outcome is. Fill in your new team members here.
We're running the show. We came up with 10 different things that might affect the growth of the plant, and now we have to pick one and do an experiment and see if it does affect the growth of the plant. And we were thinking about watering, over-watering, but she said then no, that it's positive that will kill the plant, so it's not much of an experiment.
It's kind of like the Lysol thing.
So we've got to do something else that's feasible, like maybe temperature. We could
Put it outside.
put it in the freezers over there.
That would
See how
kill
long
it.
that
Now,
lasts.
again, the question is,
That would
from,
probably kill it.
you know, the knowledge you've acquired in your lifetime, if you put something in the freezer, what do you think might happen?
Reheat it the next day.
The microwave.
Defrost it.
Okay, now don't lose that thought, okay, because could there be an experiment you did based on just what you were saying? Can you think of something? We got all sorts of things.
Because
Yeah.
if you have fertilizers, wouldn't that work? Like just having normal soil and then having fertilized soil. Leave that.
But don't lose your thought.
Oh, don't
What
lose your
thought?
thought.
When we were talking about freezers,
Yeah, what?
is there an experiment you could do?
Like
Freezer overnight?
Okay, I see what you're doing. So maybe like no refrigeration, then freezing it, and then just seeing kind of the difference. I don't know what you're going.
But could you do something to the seeds to see if it affected them?
Slow them down.
I've just affected them in some way or another. What could you do? With what you were just saying, what she was saying, add those two thoughts together.
The fertilizer affects it?
Seed refrigeration affects the seeds, slows them down.
Yes, but how would you physically go about doing that?
Put it in the freezer? For a
Did you
day?
watch it?
For what?
For a day.
For a day?
That's what we're talking about, like overnight.
And didn't have another
Is that, okay, let's think about that, and let's think about what seeds have to deal with out in the environment.
It's quite cool.
Okay, so then play environment. What would you do to do that? Try to simulate an experiment that might happen to a normal seed in the environment.
Like during the day, make it warm, and then at
And
night.
that's too complicated.
Oh, of course it is.
It is. This is something very simple. Think about what you can do.
Just put it in there for just a class period.
Well, what happens in nature?
It gets cold overnight.
Okay, and so is that just the period of time of the class time?
No.
Okay, well then, let's go.
Jen, say it.
Put these brains together.
Say something.
Put the brains together here. Yeah, but don't get off of that thought. It's a good thought. It's a good idea.
Do you have any nitrogen fertilizers?
There's lots of different fertilizers over there, liquid.
Maybe kind of seeing what the presence and absence of nitrogen in the soil.
I know your dependent variable is your outcome. So your independent variable
What's your independent variable in the experiment? What have you chosen to do?
plants. We're gonna light
Light, sure. Light is your independent variable. Okay, now the tricky part is the dependent variable.
will it grow?
So you're going to have to determine that. The independent variable is the orange juice, because what's water? Is the control. Aha.
So then the dependent is the plant?
That's the tricky part. What was the dependent variable in the Pillbug experiment? What was on that X or the Y axis? What
time?
Look at it as your general experiment.
No.
We don't have our methods.
Back to lab 2. I need one answer per team. Looking at the cycle of Brassica rapa, very carefully now, starting day 1, what are some dependent variables that you can use in the experiment of this growth and development of Brassica rapa? And this time I'll start with Team D. the height. Okay, well, that's what a whole lot of people have told me. But is that the only thing? And sometimes the trouble with height is it can be very variable. Think about grasses. Think about planting grass here in our area versus Arizona in the desert. Is there going to be a variation in height? Well, there sure are. Right? There's sure going to be. And some plants will grow so much taller if they have the proper environmental factors. So that's just one, okay? Okay, F, Team F, can you come up with another dependent variable?
With the development of the roots and then how they.
Oh, the development of the root. So we could look at root. What could we look about root?
The grow, like how it, how it grows and they are.
Remember, I have to think about what I can measure. What can I measure when it comes to roots? Okay, I'm on to Team B. A dependent variable.
The seeds, maybe?
Seeds? What about the seeds?
See how fast they grow, see how long it takes, see how large they get, see how many grows on each of the flowers or something.
Oh, you're talking about seeds at the end here?
Yeah, at the end.
Okay, okay, I thought you were here.
Oh, no.
I'm sorry, I am with you though. And is what you said about these seeds, could that be applied here, day one?
It could be. You
Okay,
see how
so
many,
let's
like
go-
the ratio,
Ratio of,
reproduction.
not quite, ratio of what? What's happening here, day one? Is that reproduction, you guys?
Germination.
Germination. Oh, I'm sorry. Okay, you put in biology 303-5. I do see it.
That's an easy way to do it.
Thing. Yeah. 303, yeah, that's it. So the hit, it should go. So did you ask Nicole? Did you ask Nicole, the young lady that was completely
That's how I
reorganized?
saw that one. You can see the roots of that one.
A3 is for organization, so you're kind of reorganized.
The one that was.
Because remember the requirements of germination, they are,
Oxygen,
okay, moisture and temperature. Now what might the plastic liner cause that might create what problem?
drowning out the seeds, or like too much moisture.
Drowning out the seeds. And what did you remove of the three factors?
Or, yeah, you take oxygen out because it's ultra-low.
You took the oxygen away by, like me putting you in the swimming pool and holding you under. You would not be very happy. So that's what you did to the seed. So I would be a little careful about how you're going to set the experiment up.
Daphneas.
I'm not sure if that is natural as
They're all
you're trying to
females
keep it
who reproduce again.
like a natural
We can do a couple different food measurements also, like you can put
Well, you
a
could
little
put,
bit
yeah, that's
heavier
what I
in
was
one of
going
the
to say, is
vials.
line it with
Like we
paper
can set
towel,
it
but
up
you
with
could
like
put
four
paper
of
towel on
them
top
and pick
as
the
well.
best, like kind of as a
moisture.
preliminary experiment.
Oh, okay.
You know,
Plastic
because
bag, whatever.
it's not worth
I mean, as long as
risking
it's
all, put on
But I mean, is that a
in
suitable
one, and then
container?
just have them
Okay,
on like
great.
hardly any.
Yeah.
Just
Okay.
one with hardly
So
any.
we can have like,
Okay.
Right. A few, like 10.
Okay, so where are we? Oh, you do? I'm so excited. What are we going to do?
Well, we don't know the amount of yeast that they typically do again. We're still like working on that. It says in here, we were worried about that we wouldn't be able to find out, like we wouldn't be able to see how many offspring they would create because it would take too long.
Oh, well, but reproduction rate is?
2 to 4 days.
Yeah.
2 to 4 days. Whoa.
Check that.
Okay, so that is then a great dependent variable, right?
Some of that bacteria out of the other stuff.
Acidity on the lower side. Okay, once you're finished.
I think we'll just be like 9.9 grams.
Well, this is saying. Yeah.
Well, we'll just try this.
Yeah. Okay. Yeah, we'll get the Jack can.
Because if you're a living organism, you see, like you are, you want to be.
The title of our experiment was The Effects of Temperature on the Mass of Penicillium notatum. And our inquiry was done to address the question of and explore the effects of temperature on the increase in mass of P. notatum. And
since other similar organisms, such as mold, develop more slowly in a refrigerator than in a dark cabinet. And our data suggests that our hypothesis was confirmed because the 5 cultures placed in the cabinet at 24 degrees show significantly more mass. The unexpected result was Petri dish number 4, which according to the data seemed to lose mass. He's a little guy that kind of dipped below the line. To improve our inquiry, we would use a larger sample of Penicillium notatum and repeat the experiment using more Petri dishes so we would have more data to work with. Let's see. Oh, and we would probably use a variety of temperatures too, not just 2 temperatures, to kind of get an idea of which temperatures best propagate.
For our experiment, we wanted to answer the question, how does temperature affect the change in weight of Eisenia foetida? For our experiment, we placed 3 different groups of worms in 3 different temperatures, and at the end of the 2 weeks, we measured the worms to see how much weight they had gained. And we predicted that the worms kept at room temperature would gain the most weight over the 2 weeks because we thought that the cooler temperatures would slow down the life cycle of the worms, causing them to eat less food.
we also, by doing this experiment, we also decided to have a moist paper towel underneath the soil before we even put the soil in, just because we were kind of nervous to see like if the worms would come out, just because there was holes at the bottom, and it would also be for moisture, so they would still have moisture within the soil. Plus, we also had a wet paper towel over the containers just so they could have also more moisture, just because we weren't going to be supervising them for a long period of time.
And the conclusion that our data suggested, it supported our hypothesis that the worms kept at the warmest temperature, 25 degrees Celsius, would experience the greatest increase in weight. That was the trend, the trend that went from the lowest to the highest temperature.
And so in the room, I'm a facilitator really, not an instructor. So I'm just one of you, and I'm going to be helping you do some science. That's our kind of philosophy in the classroom. Okay, next question from Team E.
What is chemosynthesis?
What is chemosynthesis? Oh, now we need some classroom help again. Anybody have any thoughts about that? Is there any words you've heard before that are similar? Any parts of the words? Team A.
Isn't it make either create their own food or obtain food?
They can, I'm sorry, say that again for me?
They can either make their own food or like obtain food
They can make their own food or obtain food through absorption. Wait, what a good listener. Chemosynthesis. Does that make sense to people? Yeah? No? Can you give us an example of an organism that might use that method to obtain energy? Ooh, quiet. Okay, let's look at that word one more time and break it up. What word that you're familiar with sounds like chemosynthesis? Photosynthesis. Okay. So we know, what do we know about photosynthesis? I think we talked about that during Lab 1. Did we not talk about that, I think? Okay, photo means, okay, so that's your what? Source of energy. Excellent. So in chemosynthesis, what do you think chemo stands for? Chemical what?
Source of energy.
Source of energy. Say it loud.
Chemical source of energy.
Okay, you can see both on here. You figure out what's going on. There it is.
Oh my God.
Here, did we get a?
You know what, I think we probably have some of this in our last lab.
Alright, we definitely have stuff under here.
What the?
Okay, well, we've got to move into some presentation. Okay, so keep your eye on the clock, people. We have about 20 minutes to work. Yes, you do need instructions on this. Okay. This, as I said, was a micropipette. This smaller one should fit this tip, I think. No, maybe I'm wrong. Or maybe he didn't give you the right tip size. No, that's not right. This is too big. But both is not right. Is this this one? Okay, that fits that one. Okay, did you want smaller than this? Activity 1 is having us focus on the concept of growth and development, which is what our 2 open inquiries are on for phase 1 and 2. And we've already begun that, so you have some thoughts about growth and development for Razika Rapa. So we're going to do it on the piece of paper in front of us, but we're going to do it as a team instead of each student, okay? And I'm going to do it this way, and then later a team member can make it into a concept map using Inspiration software and put it in your Team Lab notebooks. Okay, so that's how I'm going to alter that one. Activity 2 is dealing with the results of your experiment. And we do have some toys in the room. We have digital cameras and video cameras, if that will help you to document the results of your experiment.
We have a lot of the same ones, like habitat, we have swimming for one.
Okay.
And just a supplier of oxygen.
A supplier of oxygen. Oh, that's quite a thought. Okay, let's take that thought for a minute. Team B, what what comes to mind when you say a supplier of oxygen?
We have photosynthesis, water is broken down to different components such as oxygen.
Oh, okay. Wow. You guys are quite a class here. All right, I have to see this. Okay, let's see, chalk. So we're talking about photosynthesis. I'll just put a P up there for photosynthesis. And what did you tell me? You said water. So here's water. And what happens to the water?
It's broken down to
It's broken down into components. Do I need something else? What else do I need here? Do I need some other things? I need carbon dioxide. Oh, okay. And anything else do I need?
sunlight.
Team A, what else do I need? I do. And what do I get over here?
Energy.
I get. Oh, I need
It will be
energy.
a source of energy, which will be the sun.
Okay, put some energy in there. All right.
Oxygen.
I get oxygen. Okay. So then animals can breathe. And what else do I get? What happens to the C up there? Can tell me, Team D. Can you tell me? Anybody from Team D? What happens to the C? I have to do something. I can't just leave it dangling. It's got to be on the right side if it's on the left side, right? Team F, do you have some thoughts? Oh, I know you have thoughts.
Turns into sugar.
It turns into sugar. Sucrose. Oh, okay. Or glucose. Okay, glucose is a little simpler than sucrose. Okay, but that's where the C gets put, right? So yes, so we do have, and it just happens to be, but not that I would expect you to know this, C6H12O6. So we are splitting water and adding the H's in water to carbon dioxide to get our food. This is what we call our food, right? Okay. Now, question for you. These are great thoughts. You guys are good thinkers this morning. Do we make water in our bodies through any of our processes? I mean, do we produce it? We're not making it. I don't mean create, but I mean, do we produce it? Yes? What do you think about that? Team A. Do you think so? Well, maybe urine. Well, that's a good thought, right? Yeah. Okay, Team D, what do you think? You don't think so? You know, you guys are business majors, a lot of you in here, so you know what products mean. Do you think we make water as a product? Team F. Yes, no? Yes, you think so. How's that happen?
I don't know. You can buy bottled water. That's like a direct product.
Well, we do need eight glasses. Our team told us that. Yeah. So, okay, let's go and talk about what we do. What do we do every day that we have to do to nourish our bodies? Eat. Yeah, we do have to eat. And so, what do we eat? Food. Yeah. Okay, we do eat food. What's in the food? What are we really eating? Sugar. We're eating sugars. And what is sugar? What is it? It can give us energy, but what is it? If I had to define it, it is starts with an M, the big M word. No, it's not a mineral. It's not a mineral. What is it? It's a molecule. Oh, that dirty word, a molecule. It is. So you eat molecules. So if somebody said you ate sugar, yeah? Then what happens when you eat sugar? What do you do? Yeah, how?
Certain things are absorbed.
Chemical process. And what do you need to break it down? Do you need some water? Yeah. Well, you probably need some water, but what's happening here? If I take a look at this, I'm eating food. What am I doing with my food? I'm burning it, right? And what am I making? What do you get rid of it?
Carbon dioxide.
And, so if you go this way on our board, which is what we do, animals, voila. So you do produce water as a product. There it is. And so we recycle water in nature as well, the living things. Kind of cool, huh? It is pretty cool. Sorry, what part of the fungi, the rhizopus, do you think it's going to interact?
You know something?
Well, we're talking about
I
eating.
got a C in biology then.
It doesn't matter what you got. You got a good brain. What part do you think is going to interact? You know the structure.
The cytoplasm.
You know the structure of the
skin?
organism. Now, does a fungus have skin?
No, it doesn't.
Okay.
But doesn't it like
Good.
it has an umbrella and then you go under the umbrella? Is that how they feed, or is that how they? I don't remember.
What did yours think? What did you find out?
Remember the movie last week? What did the movie tell us about fungi? Remember?
They're decomposers.
Okay, but how do they carry out nutrition?
Your stomach's on the outside.
Now, does this organism have a stomach? The one in that petri dish?
Yeah.
Okay, so what does it do? How does it feed? What did the movie say it was?
Which one?
We're talking about this one,
It
right?
breaks it down into smaller pieces.
Yes, it does do that. How does it do it?
With secretes.
It does secrete something.
I'm on a roll.
Oh, you are on a roll. I am very impressed. And so what does it secrete? Enzymes. We have what we call observation bias. We have trouble with that in science, don't we? Because it seems as if up there, Team D said, well, I'm not sure, maybe. Well, it's pretty tough here if you focus up and down. Are they in there, aren't they? It's hard to tell. But what do you really think? Do you think that this mold would eat yeast? What do molds like to eat from your background research? Anybody know? Oh, bread and fruit? Good, good. Those are from your experiences. You've seen where they grow. That's exactly right. So do you think they would like yeast? You think they might? Well, do they eat things that are dead or alive? Should we ask that question? Oh, nobody thought about that. Well, that's an interesting thought. So what do you think? You know, some things feed only on live. What kind of a role, an ecological role, do these organisms? They're decomposers. What does that tell us? They kind of like things maybe that are already dead.
Okay, for growth and development, there's the obvious one that for any kind of growth and development or increase in size, it would need a plant or any kind of animal or anything would need some kind of energy.
Ooh, that's good thought.
Supplied by nutrients.
Okay, hold up on energy. What
What's
kind
that?
of energy have we learned? What kind of energy do you need to grow and develop? Ooh, during respiration. And can I have an adjective to respiration during? Okay, aerobic. I was thinking of another one. Can anybody give me another one? Well, I'll ask it in this way. Is the respiration you just identified the, oh, I need more air or not? What is it? It is? Needs an adjective. Where are my English names? What kind of respiration? Somebody said aerobic. Where does it take place?
Cellular respiration.
Who said that? Excellent. If the apple juice was at a 4.5 pH, right, and the lactobacillus seems to like 4.5 to 6.5, why do you think it did not grow on the apple juice?
Well, like we had said in our conclusion, just that the standard one, just the normal agar, was more nutrient rich.
Okay, so can you tell us what you meant by standard agar? What was in that versus what was in the apple juice agar, what you called apple juice agar?
The standard agar is made especially for growing bacteria like this, and it includes like nutrients that the different bacteria will prefer. The apple juice agar is like apple juice mixed with like a gelatin almost, so it's basically just the apple juice itself, no other extra nutrients or anything.
Okay, so that says to us, that evidence says, what does that say? That the lactobacillus
Needs the nutrients that were included in the agar probably to grow better.
exactly.
We just have those two questions in the auction. Is there anything else that anyone has problems with?
Facultative.
Aren't like anaerobic.
That's body structure.
No, I
mean, bound,
No, I don't think so.
infections.
With it, like, kind of. This is for Snorri. Great.
Maybe we could get like a dissecting instance. Since we need to use both a dissecting and a compound microscope.
Trying not to open.
You guys got that?
Red dots around it, and round, just blob in the middle. I don't think it's eaten. Just what you saw.
Yeah, but it's not really in focus that well.
Okay, we had the Hydra. The name of the kingdom is Animalia. It can be found mostly in fresh water. They reproduce mostly by budding, but also sexually. They show heterotrophic nutrition. Cells make energy by aerobic respiration. Cells are described as eukaryotic, and the body structure can be described as simple multicellular. They have the ecological role of consumer, and we had it includes hydros.
Okay, but you can give us another example, can't you? Yes? Oh, come on, from this kingdom? Us. Oh, yeah, okay, sure, okay. Now, Team AB today, you're together. Do you have any comments on the information presented? I'm sorry, say that one again?
Asexual because
Can you answer her question? She said she thought that they reproduce both sexually and asexually?
The budding is the asexual process.
Very good. Budding is asexual. That's exactly right. And she was thinking right as well. Very good. Anybody else a comment on the template here? Information template? Okay, we ready for evidence? What are we looking at here? Yeah, tell us. You guys that are experts on Hydra. What's that?
The tentacles.
It's a tentacle, right. Somebody called it the arms, but it's not really an arm. It looks kind of like that, though. Whoops. I know, it's hard to keep. Let me try going down in magnification. Maybe that would be a better view to see, except it's hard to see the yeast. Well, what do you think here? How are you going to know? Where does it feed? You know anything about the anatomy here?
He feeds right here.
Oh, excellent. You said the indentation? Whoops, he moved. Yeah, there's a
He
mouth
uses
there.
his arms to, or his tentacles to kind of bring stuff in.
Oh, to gather? So do you think, it's hard, it really is hard to keep these at the right place. So what do you guys think? Yeah, how do we know for sure? You really see something in there? Or you can see red pretty good here, red and green, Christmas time here. What do you think? Yes or no? You think so?
Earlier we felt like he was because he kept bringing stuff in.
Yeah. Well, do we have any real evidence though? I mean, okay, if this is the mouth, then what do you think happens?
Down.
Yeah, it goes down in there. There's actually a cavity they call gastrovascular. Just some more names. But so do you think it's really putting? Whoops.
Like a big clump.
You think so?
I think it takes time is what I found. I have a question about that.
Oh, he's not happy, but we're not making him happy here right now. He's all bunched up. So yeah, what do you think? Do you think he really is? This one is really hard.
Because what I found in my research is that on his ectodermal layer, that there's like poison, contains poison. He paralyzes the animals or whatever he eats for a little bit, then he engulfs them or he lets them digest near his skin.
Okay. So if he has something in his tentacles that paralyzes his prey, what kind of prey do you think he likes? Alive. So what do you think about that? Do you think it's going to prefer yeast for a food? You don't think so? This is a this is a tough one visually to call. I think Hydra prefers these little critters called Daphnia because it captures them and stuffs them in its mouth. But it is very difficult taking a look because the animal seems to collect things on its tentacles, maybe because they are kind of rough. They actually have little stinger cells in here that they, release to capture. Yeah. Well, he's probably searching for that critter that he's maybe hungry. What do we see? You get your little laser up there, guys. Okay, we know what color we're looking for here. What's the color? Aha. And now point out to us a bacillus. Where's the bacillus? Where's the bacillus? Anybody tell me? Where's the bacillus on the screen? Ah, yeah, they're rods. Look at, see? And they're prokaryotic. That's what you told us. These cells are what? You're eukaryotic. Is there a size difference? Yeah. Oh, yeah, big size difference, which is one of the differences. Because remember, these cells, you don't have a true nucleus, where here you do. Well, what do you think now? Do you think we have an eating going on here? What do you think? You guys think no now? What do you think? The presenters up there.
We thought it was.
You think what?
We thought it was when we were looking at it before.
Okay. What made you think that?
The little rock things were kind of like positioned next to the
Next
to the yeast.
Yeah, well, they're kind of dancing up there. I'm also going to give each team a critique form. Okay, you're going to be critiquing one of the teams. And on this critique form, there's also directions on to how to present. Now, critiquing in here is not for grades. That's very important. You're not grading students. You're simply evaluating them and giving them feedback so they can improve. Because that's what this course is. It's improvement. We're starting phase 1, and by phase 3, you will be an expert scientist. So this time, your role is a review team. You are reviewing somebody's work. Okay? So I just arbitrarily chose you. So A, you're going to do F. Yes.
I'd ask her because I really don't know. No, so she just makes us guess. The control, I would say just the light in general. Like, because that's what you're controlling. And then the independent would be like maybe the furthest. I can see what you mean. And the...
Okay, great. That's something you need to include when you're ready.
Our experiment, we're going to test the effects of temperature on the consumption rate of these worms. I think we're going to use apples as the food source. So we're going to cut up an equal amount, weigh out an equal amount of apples, and I think create 3 different habitats. So we'll expose each habitat to a different temperature, maybe one in the refrigerator, one at room temp, and hopefully one at an incubator at a warmer level. And we also had the idea to control the light, so the one at room temp we're going to have to cover in a way to keep it dark because the refrigerator is dark. And I don't know what the lighting condition is in the incubator, but we're going to have to keep that dark too. And we need some help with our dependent variable because we're thinking, if these worms are decomposing, then they crap out, what they eat, most of it, because it's going back into the soil. That's their role. So I don't know how we're going to measure how much they consume if they're just contributing it back into the whole composite of soil and food.
So this seems to be a puzzle for this team, but the idea is okay, don't you think so? The idea of what they're asking. So what do you think? You're thinking apples? Okay, we think we better focus in on the apples, is what Team B thought. So what can we do? Is there something we can do? How can we set this experiment up that we can look at the consumption of the apples? I think they're going to slice them up, but what do you think they should do to figure out? This is something they're going to have to figure out to show us, to produce some evidence.
Do they take pictures? Like before and after shots and something like that? See how much, or as time period
do you think the apple is going to be weighable after? You think it is going to be?
Well, yeah, we have to remember that, like she said, they might evaporate, shrivel up or whatever.
Okay, but if it's buried in the soil, the evaporation will not be too bad because we thought about boring. But what else? What else do they have to think about here?
You could start by getting like a mass of it, and then measure the mass of the apples that you're adding. Put them in there. Take another mass, and then after a while, take the mass again, to see, and maybe get like the difference there could be what you could call your consumption. You could do the difference in total mass or something. But that'd probably be skewed by growth of the worms. Like if the worms grow at all, then they're going to get bigger, and the mass is going to increase on their end. So just because the mass would be different doesn't mean it's all accounted for in the apples.
We took the worms out and measured.
Yeah.
Took the worms out and did what?
If we measured first, say we used 2 grams of apples and 10 grams of soil to start with.
And how many
Then-
grams of worms?
Oh, well, I don't know, a couple grams of worms. 2 grams of worms. But if we measured the apples and the soil, then combined them without the mass of the worms, and then like after 2 weeks, take the worms out and measure them again.
Okay, and then activity 5 is a team-to-team swap, as we have done previously, where you are editing somebody else's writing. There's a form on page 313 in the lab manual to do that. Remember, this is a really valuable way to help each other get better juvies. It's not like you're grading the team. No, you're just helping them improve their writing. Okay, it basically looks the same way. Here it's down here. Okay, now you're going to probably have to move it. I'm not sure where it is in terms of who's used the last. No, and then you will have to reset up your experiment, and that means, or you're going to repeat what you did.
Well, yeah.
Okay. Well, maybe you could do. Maybe those seeds are still okay. You can dig them up and look at them. Right.
All right, give it a little tilt.
Wait, is this one that was
Put it back on top.
What do you think? Okay, you have designated an independent variable.
Thanks.
But it sounds like it might work out to what you're trying to do. Yeah.
Lower, but higher acidity.
Oh.
Okay, so what you really have to do is start investigating some pHs. Like, what is the pH of that distilled water? I don't know. You better find that out.
It's lowering.
What is the pH?
How long does it do this for?
What is the pH of that?
Till like it
What
stop.
happens to pH when
No, I think
that's
it
by carbon?